Hello friends, welcome back to my YouTube channel that is BiologyCenter. Today I am going to teach you how to enzyme increase the rate of a particular reaction. Okay, let's see. First of all, as I was stated earlier, when talking about enzyme, then enzyme contain different kind of configuration. Okay, different kind of a structure. Okay, what kind of basically enzyme is made up of protein. Okay, so it contains different kind of configuration, different kind of a structure, that is primary structure, secondary structure, tertiary structure and quaternary structure. Okay, but when you're talking about a tertiary structure is called most biologically active structure. Why? Because it contains crevices, loops and pocket in which, okay, which provide the activeness to the enzyme, okay, in which substrate can't bind. Okay, but when the substrate bind to the enzyme, it will form a substrate enzyme complex. Please do remember, this substrate enzyme complex is called transient phenomena. Please do remember, it shows transient phenomena. It simply means it is weakly bonded. Please do remember, it is weakly bonded. Okay, this transient phenomena, that is substrate enzyme complex, is reversible. Okay, now after the formation of substrate enzyme complex, it will form another intermediate stage, that is product enzyme complex. Okay, and the followed by that is product will form and the enzyme will remain as such. Now this enzyme will ready to catalyze the next reaction. Okay, now let's see some feature of substrate enzyme complex. Okay, and talking about it, this is, as I was stated earlier, this shows a transient phenomenon, that is weakly bond. Okay, now it has very high energy. Please do remember, it has very high energy. Okay, due to which it is less stable, that is unstable. Please do remember, it is unstable. Okay, let's see in the graph. Okay, let's see the graph. First of all, y-axis represent the potential energy. Okay, please do remember, y-axis represent potential energy and x-axis represent, that is progress of reaction. Okay, progress of reaction. Okay, now as I was stated earlier, a transient state, that is substrate enzyme complex, is very high energy. Okay, so it will be, this is the transient state. Okay, transition state which has very high energy. Okay, now this is the region which is called substrate. Okay, substrate or reactant. But when you're talking about a product, then it is present here. This is product. Okay, now let's see. When you're talking about a substrate and product, then the substrate contain high energy as compared to the product. Okay, so it simply means it release the energy. Please do remember, it release the energy. Okay, due to which this kind of reaction is called exothermic reaction. Please do remember, this is exothermic reaction. Okay, now let's see. This is the region that is called activation energy with enzyme. Will you remember? And this is called activation energy without enzyme. Okay, now let's see. When talking about the transition state in terms of energy, then transition state contain highest energy, then the followed by substrate and the last one, that is product. But when you're talking about the stability, okay, then the product is the major one, then the followed by substrate, then the last one is, that is transition state. Okay, now let us see What do you mean by activation energy? Okay, it is the minimum energy of substrate which is required to cross the energy barrier, okay, and proceed the formation of product. Okay, that is called activation energy. Please do remember the minimum energy, the minimum energy, the minimum energy of substrate which required to cross the energy barrier. Please do remember energy barrier and to proceed the formation of product. Okay, so let's see, when talking about the enzyme, okay, then it simply lower the energy barrier. With enzyme, this is the region, okay, with the presence of enzyme. But when talking about with the absence of enzyme, this is the region. Okay, so let's see the role of enzyme. What are the role of enzyme? It simply lower the potential energy barrier so that more number of molecules can cross. When the more number of molecules cross this barrier, more number of product will be formed. Okay, now it simply lower the energy barrier. Okay, this is how the enzyme increase the rate of a particular reaction. Okay, now let's proceed further. Nature of enzyme action. Okay, now let's see. Nature of enzyme action. Okay, as I was stated earlier, we're talking about enzyme, then enzyme contain tertiary structure. Okay, okay, now let's see. Enzyme contain tertiary structure in which crevices and pockets are created. Okay, now these pocket and created in which substrate bind. When the substrate bind, it forms a substrate enzyme complex. Okay, this is called transient phenomena. As I was stated earlier, this is very short lived. Okay, if you remember, this is short lived. Okay, and this substrate enzyme complex is also reversible. Okay, now it has very high energy and it is very unstable, as I was stated earlier. And the next one, that is enzyme product complexes, intermediate stage also formed. Like this is another intermediate stage. Another intermediate stage. Okay, now after this formation of enzyme product complex, it will form a product and the enzyme will remain as such. Do you remember? It will remain as such. Okay, now let's see in a figure. Okay, okay, now let's see. as I was stated earlier, in a crevices and pockets substrate band. When the substrate band, then it alters the shape and size, due to which is change in their specificity. Okay, when the shape is changed, no, the bond formation takes place. Okay, no, the tightly bond substrate enzyme complex, it will tightly bond. No, when the substrate enzyme, there is a bonding, breaking of bond takes place between substrate and enzyme, and the joining, that is formation of bond, takes place between product and enzyme. And the last one, that is enzymes will remain as such and the product is formed. Please you remember, product is formed. Okay, and the enzyme remain as such. It remain as such. This is how the nature of enzyme action. Okay, now let's proceed further. What are the factor affecting the enzyme activity? Okay, let's see. Factor affecting the enzyme activity. So let's see, what are the factor? That is pH and temperature. Second one is, that is concentration of substrate. Okay, and the third one is, that is inhibitor. Okay, now let's see, when you're talking about the pH and temperature. Okay, now as you all know that all living beings, okay, all living beings, for example, let's see, talking about the human beings, then our body, that is in a narrow range. Okay, enzymes work in a narrow range, that is 37 degrees Celsius. This is the normal body temperature in which the enzymes work very efficiently. Okay, please remember, in a 37 degrees Celsius it works very efficiently. But when we increase more than 40 degrees Celsius, then the enzymes will denature. Please remember, enzymes will denature. Okay, now let's see the graph. In the same fashion, let's see the graph. In the same fashion, when we cool or freeze, okay, when we freeze, that is when we temperature, when we decrease, then it will inactivate the enzyme. Okay, inactivate the enzyme. Okay, so let's see in a graph. Okay, y-axis represents activation energy, that is temperature. This is also activation energy, and this is a pH. Okay, now this is called optimum. This is optimum. Okay, in which maximum activity takes place. Okay, now in the extreme condition, in the both case, when the temperature is high and temperature is very low, then the activity is minimum. Okay, activity is minimum. And talking about temperature, either pH, activity is very minimum. Okay, and the optimum, that is in a narrow range, the activity is very high. Okay, now talking about the concentration of substrate. Okay, now and let's see the concentration of substrate. Now, first of all, talking about concentration, then it will increase the rate of the reaction. Okay, with respect to time, it will increase the rate of reaction up to certain, okay, up to certain. Let's see, when talking about an enzyme, if the all enzyme occupied by the substrate, if all the enzyme occupied by substrate at a time, that is called saturated. It means that enzymes become saturated. Okay, no, there is no enzymes left behind. Okay, that is called saturated. It means that the rate of reaction will increase up to saturated point. Okay, then it will remain constant. Okay, now let's see in a graph. This is called velocity. Okay, and this is called substrate. Y-axis represent now let's see the concentration of a substrate up to which in which the saturation point is found, that is called Vmax. Please do remember this region is called Vmax, and the concentration of substrate in which the velocity becomes Vmax by 2. Please do remember this is called Vmax by 2. That concentration is called Km. Please do remember this is called Km. Okay, now let's see the inhibitor. Okay, when talking about inhibitor, that it resembles the structure of enzymes. Okay, see, for example, this is an inhibitor. Okay, this is an enzyme. Okay, then their shape and size are all same. Okay, now this is a, for example, this is a substrate. Okay, now they will compete to each other. Okay, they will compete to each other. Okay, now it will bind to the substrate, for example inhibitor. Okay, so that enzyme is inhibited. Okay, now for example, let's see, for example, that is succinic dehydrogenase, is inhibited by melanate, because it resembles the structure of succinate. Okay, now this is how the inhibitor works. Okay, for example, talking about a bacterial pathogen is also inhibited by penicillin. Okay, now let's see, penicillin interfere the cell wall of bacteria such that it prevents the formation of cell wall. Okay, but the content of cytoplasm increase with respect to time. It increase, and after some time it will lyse the cell wall. Okay, that is called lysis of wall. Okay, now this is how the enzyme work. Okay, Now, thank you guys for watching my video. If you do really like this video, then please subscribe the channel and share with your friends.